Hi everyone. Gives me great pleasure to introduce Marco Bertamini from University of Liverpool, Department of Psychology. Well, not really Department of Psychology. Is it Institute of
Psychological Sciences, or yeah.
Oh, okay. Marco got his studied psychology initially at the University of Padua in Italy, and then got a PhD at the University of Virginia. I think with
Dennis Profit.
Dennis Profit, right. And since 2000, he's been running the Visual Perception Lab at the University of Liverpool, where he's currently a reader. And Marco's work is very interesting because he works on a variety of different issues. And we had he was having a debate with Steve about whether to talk about mirror perception or symmetry perception. And I guess he decided on the latter. So he's going to tell us something about perception and symmetry. Thanks.
Thank you very much. Thanks for the invitation. It's very easy to start off a talk on symmetry by simply pointing out that there is interest from physicists, mathematicians, art historians, and of course psychologists in symmetry. As a psychologist, we will focus in particular on the perception of symmetry, and the starting point is to remind ourselves of the fact that detection of symmetry is fast, accurate, and robust. I'm not going to review the evidence for that. We're going to take that as a starting point today. There are a couple of review papers here for you. But we know for a fact that you could say the visual system is tuned to visual symmetry, and it's good at processing and extracting and therefore responding to visual symmetry. So next question, general question, is to ask why, and I will mention three ideas. I will mention three ideas which are present in the literature, and we will then move on to the actual topics, which will be more about what do we know about the brain processing of visual symmetry. But just starting off with these ideas, the first one is quite a popular one, and you would have seen this, I'm sure, and read about this in many places, including beyond just the scientific literature, and that is the idea that the degree of deviation from symmetry gives you some idea about the fact that that individual might be either in poor health or something wrong or not quite right with the genetics as well. So that is because normal development produces symmetrical bodies, so deviations are a sign of something that has gone wrong. Therefore, you might want to select, in mate selection, individuals which are more symmetrical. These are just some of the species in which a preference for symmetrical mates has been documented. As I said, this is a very popular idea. It's almost, however, there is a bit of a mismatch between how everybody know about this idea and the fact that biologists are to some extent rather skeptical about this. There are limitations to how far you can go with this argument in terms of the signal to noise, in terms of actual some of these studies are difficult to replicate, and also sometimes when people have tried to look at the correlation between degree of symmetry in individuals and their health or other indices of quality, the correlations aren't always so clear. So the next idea, which has been put forward also a long time ago, and also in part from the field of evolutionary biology, is that over time, this is learning both at the epigenetic and evolutionary terms, you might develop a sensitivity to symmetry simply because that is the average, let's say, stimulus. If you're exposed to a large number of stimuli, they all vary because of a large number of factors and therefore randomly in all possible direction. The more typical central tendency will be symmetrical. You can demonstrate that with artificial neural networks, but also with some animal work. For example, like this one study here, as I mentioned, in which if you have chicken that are trained to respond to stimuli that are skewed to the right and also stimuli that are skewed to the left, they will then happily respond to symmetrical stimuli, which are stimuli they've never seen before and have not been reinforced for. So this idea is that preference for symmetry or sensitivity to symmetry may be a byproduct of learning. Another way in which it could be a byproduct, which I'm going to put on the same slide to keep things compact, is that perhaps also the fact that our brain is, or brain of vertebrates has a degree of bilateral symmetry may also be the origin of sensitivity to symmetry. Finally, the third idea is that symmetry actually has a functional role. There is a usefulness in extracting symmetry because it is linked with the presence of an object, and therefore it may help in image segmentation, in figure ground, and in finding and representing objects. So starting with the point that Mark made a very long time ago, those black figures actually come from Mark's book. In his examples, he was pointing out, that mathematically symmetry is about self-similarity, rigid transformations. So those are three rigid transformations that you could say they are equivalent in that mathematical sense. But for perception, the first one is very different from the others. That bilateral symmetry is easily appreciated without effort, Marx pointed out, and that has been confirmed then by many, many other studies. So why is that special? Well, it perhaps, as I was saying, is special because, as the Gestalt School of Psychology also proposed, is a principle of organization. You will use bilateral symmetry as a way of organizing and segmenting and doing figure ground organization because the bilateral symmetry is associated with with presence of objects. And there is some evidence to support that. Perhaps you can even go farther and say that it might help recovering 3D structure from shapes, from the projection of three-dimensional objects, or help find objects in a contour integration task. I don't know if you can see the contours on the right. In that study, they compared performance in detecting closed contours which were either symmetrical or not, and they found a small advantage for the symmetrical contours. However, there are limitations to this story as well. Even the last effect about contour integration, the effect size was quite small, as the authors pointed out, and did not extend as soon as they moved those contours away from central vision in the periphery. So we have, if anything, we have no shortage of explanations of why the visual system should be sensitive to symmetry. It's not that we don't have an explanation, we perhaps have too many. And part of the problem is that these explanations are not mutually exclusive by any means. They could all be true in some degree, all of them, and therefore all be combined. As I said, we'll use those as background, and because it's difficult to remember all of them, I will use to refer to them some keywords. So the first one about evolution and mate selection we can refer to as sex, and the second one as byproduct, and the third one as figure ground. So it's going to be sex, byproduct, and figure ground, which is not quite sex, drugs, and rock and roll, but it should still be an easy way of remembering them. Now I am going to, after that introduction, I'm going to introduce the more substantial part of the talk, in the sense that, well, this is still background, but it's background specifically on what we know about what the visual system does in response to the presence of symmetry in a stimulus. And a good starting point So this is a lot of progress has been made on this in the last 10 years or so. A good starting point are fMRI studies because, as you can see in this slide, we can get a nice map that already gives us an idea of the areas that are involved. And two clear things have emerged from these studies. The first one was that there wasn't really any major role or involvement of V1. And the second was that there wasn't a single area that stood out as being an area that you could say this is the symmetry-sensitive area. As you can see, there was a whole range of areas, extrastriate areas, mainly extending to the LOC. So a ventral pathway, but a fairly extended one. And although these pictures come from one specific study, this has been confirmed in more than one study, and we are pretty confident about this pattern. So fMRI has pointed to these extrastriated visual areas and this network, and other techniques have found converging evidence. So TMS studies have found that disruption of disruption of the function of the LOC will impair symmetry detection, and EEG studies have found a particular ERP and negativity, which I will show you in the next slide, starting from 250 milliseconds, and as the word sustain says, then lasting for a long time. And all of this evidence is part of a single story that has provided us with evidence that there is a network sensitive to symmetry in these extrastriate areas, and that, as we will see, will always respond to presence of symmetry in the stimulus. So given that it's relatively easy to record this type of response, the next step is to try and answer some additional questions using, in particular, I will focus on the ERPs. So this is the slide that will look complicated to some of you and trivial to others. Problem is, depends how familiar you are with ERPs. What you see in the top, I have a pointer here, so I might as well use it. So here you see a typical ERP. 0 is when the stimulus comes on the screen. You have P1, N1, and then these waves here are shown in two different colors for when the stimulus was a reflection, therefore symmetrical, and when it was random. Well, you see some examples of the stimuli here, but they're not important at this point. You'll see other stimuli later. The important thing to notice is that the wave for symmetry is lower, is below the wave for random. So that's why we talk about a negativity. It's negative in relative terms. It's lower than the random wave. You can subtract the two, and you will see this posterior negativity plotted like this, and I will use sometimes that type of plot and sometimes this one. These electrodes here are on posterior regions, and as far as we can tell from source localizations, they're picking up activities that comes exactly from those areas that you've seen from fMRI. So far we have a signal, and it's quite strong. It's a nice strong signal. Is sustained in the sense that it's not only present in a narrow window of time, and is negative because this negativity associated with symmetry. So this is our signal, and now let's try and use this to address I've put 5 questions here for today's talk, which are, I think, quite interesting. The first one is, symmetry more typically we associate symmetry, the word symmetry, with bilateral symmetry, but technically could mean a lot more than that. So the first question is bilateral reflection, which is the symmetry of, say, human faces, for example, is that special? Mark had already pointed out that it was special. We know from work on sensitivity and perception and psychophysics that it is special in the sense that it is the more salient. But how does that correspond to the neural response? Is it from different areas? Has it got a different time course, and so on? So obviously that would be quite interesting, because some of those ideas I mentioned do give bilateral symmetry a very special status. If you want to select for the best mates, then you are talking about bilateral symmetry fundamentally. If the attractiveness of a face, instead of being based on bilateral symmetry, were to be based on rotational symmetry, you would be in trouble. That wouldn't be a good strategy, right? Next question is whether all of these measures are automatic, in the sense of something which is generated by the information in the stimulus independently of the task. The third one is, remember I mentioned the fact that perhaps this sensitivity to symmetry may have to do with the fact that our brain is bilaterally symmetrical. So how important are the connections between the two hemispheres? The fourth one is, how do we go from symmetry in the image to symmetry which is independent of view angle, therefore moving from the image to an object-based symmetry, which would be more important for most tasks. And finally, how does all these measures that we have relate to, going back to the whole Gestalt idea of goodness? I will say more about what I mean by goodness. But for now, intuitively, it's a term that has been used in perception to refer to how salient something is. In the original literature, it wasn't defined very well, but there is progress on that as well, as you will see. Okay, I will take actually the first 2 points together. I think I have some data that will be able to, which I can use to address both of these questions. Is bilateral reflection special, and is this response automatic? Oh, I can just use this. Okay. Okay, so this is to show you some type of abstract symmetry that we've used. In all of my work, we've focused on symmetry in abstract patterns that have no associations with familiar objects or anything like that. There's a big advantage in the study of symmetry in that we can generate an infinite number of these patterns. They don't need to be familiar, and they never need to be repeated. So our participant never see the same pattern twice, because you can always generate as many of these as you like. Here you see them being made up of a relatively small number of dots, but there is nothing special about these patterns. What you will see across the talk is patterns still abstract, but made with lines, contours, with high density dots, and so on. A very good thing is that the story does not depend on your choice of how you make your patterns, as far as we can tell. The next interesting thing is to say, so what do we mean by symmetry? Well, we mean a particular relationship between one part of the configuration. In this case is the left and the right halves. Okay, so if you look at these two halves of the configurations, they are related to each other. So that's what symmetry is the rigid transformation that matches the two. And to make that point even clearer, you could just look at half of these patterns And they're all generated in the same way. In other words, these are just examples of our patterns, but they're all the same. They're all generated by the same algorithm. If you only look at half of them, you'll never be able to tell whether it's a reflection, a rotation, a translation, or even a random pattern. What makes the categories, what creates the categories, is how one half relates to the other. Okay, so it's all about correspondence between parts. At the bottom here, you see that there are really two experiments here with two groups of people, the same patterns, but for some people, they had to report the category. Other people only reported oddball trials in which there were some square all of a sudden. And those were rare events which were then excluded from the analysis. Okay, so in one case, in the second case, symmetry was never mentioned at all. Okay, so we have random, which is the solid blue line at the top. As you see, the others are all below, so they're all negative. And reflection is the most negative. That is true for the group that discriminated symmetry. Is also true for the group of people who only did the oddball detection. So the answer to the question of is bilateral symmetry special, unfortunately, sometimes in science the answer is yes and no, meaning yes, it's special because it's the one that gives you the largest amplitude. If you subtract these two, the green is lower than the others, but it has exactly the same time course, exactly the same onset, and the same shape. So it looks as if bilateral symmetry is the optimal stimulus for this network, but the response, other than that, looks very similar. Okay. That was the answer to the first question. The answer to the second question is that this response is automatic, and the task in this data I've shown you, but in other experiments in which we've used different tasks, the task makes very little difference. You get the same neural response to symmetry. What matters is what's in front of you on the stimulus. That's consistent with the fMRI work as well, in which they found the same thing. So this is really an automatic response where the visual system can help to produce this activation in response of presence of symmetry in the image. Okay. The next question is going back to this idea, which is quite an old one in the literature. Perhaps the fact that bilateral symmetrical stimulus is activating anatomically corresponding parts on the left and on the right side, that's something that it could explain why bilateral symmetry is more salient. There was, again, this literature is a bit old, but in the 90s they did have a couple of patients which were born without corpus callosum, and they had struggled seeing symmetry. So that gave rise to this callosal hypothesis that the connections between the two hemispheres were important. I can just press this. Well, one simple way to check that is by checking how important is the vertical axis as opposed to an horizontal axis, because the whole argument about corresponding locations in the two hemispheres is specific to a vertical axis. It's true that in general vertical axis is the most salient. Again, you can go back to Mach and you'd already noticed that as well. However, the advantage for the vertical is not a very robust effect. You can prime people for where the axis is going to be, and the effect pretty much disappears. So in this experiment here, we had, there is a line there, I don't know if you can see it. So the line was on the screen before the pattern. They knew where the what the orientation was going to be, and therefore their performance was not, behaviorally, was not significantly different for vertical and for horizontal. And not surprisingly, therefore, there was nothing different fundamentally between vertical and horizontal. So that's a simple answer to the callosal hypothesis, which in any case is not very popular any longer anyway. But we can go beyond that. So let's go all the way to presentation of stimuli that are entirely in one hemifield. So sorry about the red. Can you see the red? So there are red stimuli here. And there is a white fixation mark. So you're fixating here, and you always have two patterns, one on the right and one on the left. You're not moving your eyes, and we know that you're maintaining fixation. So I like this experiment because we're now going to analyze trials in which there is always symmetry and there is always random in every trial. That is, every trial has just as much symmetry as any other trial. Okay, so if you were to just take an overall response, then you wouldn't expect any difference. But the difference is that sometimes the symmetry is on the right and sometimes it's on the left. Okay, and what you see here is the same ERPs that you've seen before, but these are just the electrodes on the left hemisphere, and those are just the electrodes on the right hemisphere. So just to try and just make this a little bit clearer, I'm not going to pretend that there was only the stimulus on the right, okay? So this is just the one that is on the right side in the right hemifield and therefore projecting to the left hemisphere. And now the green and the blue lines correspond to random, which is that one, and symmetry, which is this one, because that is what was in that hemifield. And the SPN, the sustained posterior negativity, is present within just that hemisphere. Equally, you go to the other side, the same thing happens. There are some, I'm not going to go into hemispheric differences in terms of degree of sensitivity. That's a story I've left out, but it's a relatively, it's a minor modulation of this overall pattern. Okay, we have also experiments in which on the other side there was, in this case, the other side always had the other pattern. We also done it with nothing or with the same. Actually, what's in the other hemisphere, somewhat surprisingly, made very little difference. It's as if one hemisphere was producing that response entirely on the basis of what was presented in that hemifield, with very little influence from the other side. Well, no influence that we could see. Okay, so the answer to the question number 3 is that no, the fact that the brain is symmetrical is not important. Both hemispheres show this response, and they can work pretty independently. Number 4 is this response independently of view angle. Well, as you can imagine, this is important if you want to relate it to any kind of real life situations, and also interpretations on the basis of how does this relate to, say, mate selection or figure ground. Because most symmetrical objects will not appear symmetrical in the image from a generic viewpoint. Here's an example from one car that comes from not from a paper. But you can imagine a human face, and the same point would apply. Human face would rarely appear symmetrical, even though it is a symmetrical object. So everything that we've seen so far, is it in response to the information in the image or the symmetry of the objects after you've taken viewpoint into account? Okay, I'm showing you some stimuli, again, slightly different. The good news is you don't need to worry about the fact that density and other properties seems to vary from one experiment to the other. In this case, we have the important manipulation is that these patterns, which sometimes are symmetrical and sometimes are random, are presented either in a plane which was had zero slant, was in the frontal parallel plane, or with a 50 degree slant. So notice like the difference in the image is large, this and that. They're very, very different as stimuli, right? The next thing to notice is, will become important, is that our stimuli half of the time were dark red and half of the time were light red. This will become important because this manipulation allows us to have two tasks. One task in which, for those stimuli, people have to report whether it was symmetrical or not, and another group of people instead only had to say, was it dark red or light red? Light red. Okay, first, for the people who discriminated regularity, we found something which is quite a clear-cut answer. There was complete view invariance. There is no difference whatsoever in the response to symmetry, even though we are talking about stimuli which are very different from each other. This stimulus and that one, they're very, very different in terms of their images, but the response is identical. Okay, well, you could say, fine, these brain areas are so clever that they're going to respond to the degree of symmetry in the object, taking care of any distortions due to viewpoint. But that's not actually true. That's not the whole story. So remember, so far I said this is a response to the symmetry present in the image. Well, that is still true. If people are not trying to extract symmetry and they're only reporting the color, then they don't have view invariance. And the response is what you would expect if what they're responding to is the amount of symmetry present in the image. Okay? So what I'm saying is that when attending to regularity, symmetry network are view invariance, but when attending to other dimensions, symmetry networks are activated by whatever symmetry is present in the image. So achieving view invariance takes work. That is not automatic. Okay? So this is not at odds with what I said earlier. It is still true that the response to symmetry is automatic, and it is a response to whatever is the symmetry present in the image. But if you want, you can go beyond, but that is not automatic. If you want to go beyond what's in the image, that extra compensation for view will take some work and presumably attention. Okay, final point is the neural response to symmetry response to goodness. Well, what's this goodness? Well, we like to be able to say that some patterns are more salient, better organized than others. And in psychology, we struggle for a long time about trying to quantify goodness and say, how can we measure it? What we mean by goodness is some kind of strength or salience of a pattern. It's a term that comes from Gestalt psychology. And you could just say, well, it has to relate to how easy it is to see something. So you can measure it with reaction time, perhaps, or other measures of sensitivity. Okay, well, yes, that's true. And what that will tell you is, for example, that reflection is more salient than repetition or rotations. We've already seen all of that. But there is one model that gives us a number for goodness, okay? And we're going to test that. It comes from the work of Peter van der Helm and Löwenberg, and it's called the holographic weight of evidence model. The key idea is that the goodness is measured by this W, with this weight of evidence, and that weight of evidence is the amount of regularity, which the theory does say exactly how to measure, divided by total amount of information. Okay, so in that sense, it's a weight of evidence. So the good thing is that they provide formulas to compute different W for different patterns. For reflection, the model simply says that the regularity is the number of pairs and the total information is the number of dots. So every time that you have a single axis bilateral reflection, you have twice as many dots as pairs, of course. So your W will always be 0.5. Okay, so that already makes a very good, interesting prediction. It says that the goodness of this pattern is the same as the goodness of that pattern. That actually is consistent with the behavioral data. The sensitivity to bilateral symmetry does not depend on how many dots you have. It's pretty stable. But for repetition, it's a very different story. Because now the regularity is coded as number of repeats minus 1, then how many, and number of dots obviously varies from here to here, but number of repeats doesn't. So the goodness of that is much higher than the goodness of this one. And you should be able to see that that's obviously a repetition, but here is messier. On the other hand, if you increase the number of repeats, keeping the same number of dots as I've showed down here, then that W will go up. And this is a very easy to see pattern. So goodness is high here and there, but it goes down as you increase the number of dots. So the important point is that there is some behavioral pattern in our sensitivity to different symmetries and to how they are generated, which is picked up by this model. And it is reasonably good at predicting the psychophysical results, like in this case, minimum amount of time necessary to see the regularity. Okay, it correlated with the W value. Okay, so far so good. So what about the neural response? As we can measure it using this sustained posterior negativity, how does that relate to W? Well, go back to the first type of patterns you've seen. Is a single reflection, therefore 0.5, is much lower W for rotation and for translation. Well, that more or less matches. Certainly, as we already seen, the greatest amplitude is for reflection, so that is consistent. Well, we can try and be more precise. So, for example, we can vary percentage of dots that are reflected in this pattern here from 100% down to 20%. 20% is almost hard to see that there is a symmetric pattern there. And the amplitude scales very nicely with the percentage of matching dots. Now, here, I don't expect you to look at all the panels. All I'm saying is that we've looked at this across many different stimuli and different manipulations. And the key is that the key results over there is that the W always is consistent with the amplitude of the SPN. This is all of that put into a single correlation, and is pretty good. And one final point, I'm not keeping track on time, but one final point. There is another additional good thing about the weight of evidence model. It is the only model that we have that explains the effect of number of axes on the salience of symmetry. So what do I mean by number of axes? Well, when you have a reflection, you could have a single axis, two axes, or how many axes you want. And as you increase the number of axes, it does get a little bit easier to see the regularity, but not in a simple, in fact, not in a monotonic way. And that's sort of surprising, but it's been shown in a few studies now. As you can see here, in terms of reaction time, this is something we did in our own lab, but as I say, we didn't discover this. There are some dips that corresponds to the odd number of axes, okay? And that is predicted by van der Helm's model if you apply the formulas. And so no other model predicts this up and down pattern. And the next question, of course, is what will happen to the neural response to symmetry? Will it behave in a sort of monotonic increase with number of axes, or will it have this characteristics pattern where the odd number of axes have a handicap, you could say? Well, so far, okay, here again is one of those results where the answer is it depends, okay? But I think in an interesting way. So far, I've been always showing you the sustained negativity as a very straightforward, simple difference between two waves, which lasts a long time, but is fairly constant. However, if you'd paid attention, you probably noticed that it wasn't always staying exactly the same in amplitude. But until now, until this study, we'd not paid attention to that, and we considered that as a single signal and as a fixed effect, which we called SPN. But then when we went and tried to see whether the holographic model was in agreement with the amplitude of the SPN, we realized that it really depended on where we were looking along the time axis. So if we use this peak, I know at this stage seems somewhat arbitrary, but you can all see that there is a peak here which we can agree on. If we measure the amplitude at this early stage, then the amplitude of the SPN is given by the green line. And then obviously we had to do some normalizations to try and plot this on the same scale, but the W score is the red line. The blue line is another model which simply predicts that the sensitivity should go up with number of axes, and that of course doesn't have this up and down pattern. Well, in these early windows, it's amazing how useful the holographic model is. It predicts an up and down pattern which exactly pretty closely matches what we've found. However, later on, it flips and it becomes exactly the opposite story. Now the holographic model doesn't match the amplitude of the SPN. So what I said is the answer is it depends. Now we have to, we think we're justified in thinking that we have to split the SPN in an early and a late stage. And we're still working on trying to find the exact meaning of those two, both in terms of what they correspond anatomically and what they correspond functionally. So that is ongoing work. So the last couple of slides, I think the very last slide actually is just a summary of what I said about these questions and how we were able to answer them. Is bilateral reflection special? Not in the sense of being unique. All the evidence, not just our own evidence, is always the case that bilateral symmetry stands out as the most salient, as Mach had noted more than a century ago. But there is nothing that identifies a unique area or a unique time course or a unique onset of the response to bilateral symmetry. So this network is better described as a network that is sensitive to regularity in a more general sense, of which bilateral symmetry is simply the optimal stimulus. Is this response automatic? I think we can answer with a clear yes to that. What counts is the regularity present in the image. Other tasks have been used. I mean, I've shown you data with the oddball detection and with the color discrimination. Other tasks have also been used. Also, some other labs were interested, for example, in aesthetic judgments and things like that. Are connection between the hemisphere especially important for symmetry perception? Well, clearly not. On the contrary, there is there was a very clear-cut answer there where the activation in the two hemispheres was pretty much independent. Is the response to symmetry independent of view angle? Well, it can be independent of view angle, but that independence of view angle is not something that happens automatically. And finally, how does that relate to goodness? Well, actually, we found the model that comes from Peter van der Helm and Leeuwenberg. We found it very, very useful. Certainly, within at least an early response to visual symmetry, this W score has a very good match with the neural response that we can measure. Thank you.
Questions? Thank you very much, Marco.
Yeah, about the view independence part of the talk. I was just wondering if that's more to do with how you process the image. So objects in the world that are symmetrical, like faces, are processed a lot more holistically than your part-based. Your images are very part-based. And the question that you asked could have just simply been more to do with how they view the object. So what I'm getting at, is there any data to show actual objects and symmetry perception from different points of view as well? And if you ask the same questions to that, would you still find the same results?
That's a very interesting point. I think there is, though, a similarity or an overlap between when you say holistic processing and when I'm talking about asking a question of judging regularity in the objects. I know there were only flat surfaces, but they were framed. You could think of them as a object. I know it's not a human face, but yes, I would predict a similar pattern if the task is to focus on the holistic properties. Then you would find sensitivity to symmetry, but that's exactly the same story. That is, you can do it. It doesn't. It's not the same as the automatic response to the information in the image.
I might remember it wrong. I think in one of the first slides you told us that vertical symmetry is more salient than horizontal symmetry. How does that match with the W goodness? Because that would be the same for both stimuli.
No, the W would be the same for horizontal and for vertical. But remember what I said is that, Mach had pointed out already that vertical was special. The bilateral symmetry around the vertical axis was the one that was most salient. However, since then, many studies have looked at this, and although it is true that the vertical axis has that it is better than other axes. To a large extent, that can be it's an expectation effect that you can eliminate. So priming people, as I said, for example, about the axis will almost completely eliminate the advantage for the vertical axis. What I'm saying is that you can find that vertical is better, but but that seems a result of something of a separate process from the basic sensitivity to the symmetry. Does that make any sense? Yeah.
I was just wondering if that could be linked to your attention thing as well, that you maybe attend along one axis initially, and then, so if you give people time,
yeah,
that you'd get equivalent neural responses. Could it be the same distinction, really?
So we haven't systematically compared the amplitude of this neural response for different axes. My prediction would be that across, although it could be sensitive to some expectations, in general, you would find that you find the same response to all axis orientations, because the regularity is the same and it would be measured as being the same, but the W score as well. So that would be my prediction.
Any other questions?
The question relating to point 4, the question 4 that you had, which was about how if you're attending to the symmetry, you can kind of activate the sustained posterior in ways that you would expect. Now, and you're almost certainly figuring out, I'm not a perception researcher, so I'm going to use all sorts of wrong terms here, so do try and bear with me. I'm wondering if the stimuli that you're using are kind of sparse in that you're just presenting the thing that you are judging for symmetry. If you were to nest those symmetry in those symmetrical images slanted in scenes where the symmetry was incongruent with what you might expect of the play, how things will move away from you and so on, would you still be able to turn on this symmetry?
That is a very good question. In fact, we had planned this experiment also with the frame not consistent with the, as you just mentioned, the frame here is consistent with the length of the dots, but it could be inconsistent. Well, we never eventually, maybe we should, but we never ran that study because we thought, well, yeah. It's bound to mess it up a little bit, simply because now you have two inconsistent sources of information. So we weren't sure that we would have learned anything conclusive from that. But yeah, so but you're right. You have two. Well, you have a stimulus made up of two parts here. One is the black frame, and one is the dots. If you decouple them, you can create conflicts, but that would be a little bit like just having more of those red dots and then having some with one slant and some with the other. So you would end up with two patterns that might segregate or not, and so I think it's your thought is exactly on the kind of manipulation that one could do. But so far we couldn't think of good reasons why we would run that experiment.
Yeah.
Mine is actually linked with this. I was just wondering how, if instead of using this kind of stimulus, because what you're thinking is how it's going to be in depth, what if you would actually try rendering three-dimensional object, or even showing, some kind of stereoscopically three-dimensional object, rather than that transfer.
Okay, so I take the point that all of these stimuli are very simple. Okay, and it's true. By design, I would say, we so far we focused on patterns that are 2D. The density actually we varied quite a bit. Some of them are actually fairly high density, but there is no 3D shading or 3D stereo information, as you were saying. So those additional informations could be added, yes, and you could create certainly richer stimuli. But I would expect that what's true for the 2D would generalize insofar that if the three-dimensional object projects a symmetric image, I would expect an automatic activation of this network. If the three-dimensional object is clearly three-dimensional but does not project a three-dimensional does not project a symmetrical image because it's a face seen from the side or whatever, then it should, something more similar to the slanted dots should happen. So that's the prediction, but you're right. Maybe if you get them to be really realistic, then the network will be activated automatically. Then I don't know.
I had one last question before we go to the wine reception. I hope there's wine out there. I'm assuming there is. It seems like it's really nice, clean response that I'm surprised, it looks like it's a really powerful tool to explore a lot of things in shape and space perception. I was thinking one thing, just another annoying, more complex stimulus question. The fact that in face perception, the lack of symmetry is something that is, there's a difference between seeing symmetry, seeing a lack of symmetry in something that's already recognized as a face versus a stimulus that's, say, a random set of dots. The sort of the categorical or the phenomenal shift from seeing something that's sort of symmetrical to something symmetrical is not as probably as intense as seeing a face that is, like you showed us in the very first slide.
Yeah.
So I'm just wondering whether not in the very first slide where you showed us a distorted face that didn't have.
I see.
I'm just wondering what you might predict the distinctions would be between your negativity comparing a face that we should expect to be symmetrical, but it isn't, compared to one that is symmetrical. Would you expect that difference to be much larger than you're finding in going from, say, random dots to a symmetric image?
So I don't know what I can say, which is so to stress the positives. Okay, it is true. It's a clear, strong signal, easy to record. So that's a very good starting point. And also, as you've seen, okay, they were all simple, but you have low density, high density, color, no color, grayscales, sharp angles, rounded. And more. We had closed contours, for example. And across all of that, we never found any particularly interesting difference. You always get the same time course and the same negativity for symmetry with respect to random. We take that as a positive in the sense that there is a sensitivity to the regularity. This regularity can come from. Well, it has to come from the image, but the image could be colored or not colored or very different, but that doesn't matter.
Okay, so you're suggesting that it is a fairly low-level imagistic signal that is causing it, and it's not.
It looks like that. It looks like that. Remember, the fMRI studies found the same thing, that the task didn't make any difference.
Okay. Great. Well, thank you very much again.
Thank you.
I think we have a wine reception next.